Greetings, I'm Kevin Hackett and work at the USDA Agricultural Research Service in Maryland, USA. I'm one of the co-chairs of the i5K initiative to sequence the genomes of 5,000 arthropods, insects, ticks, mites, and spiders, and amphipods. The other co-chairs are Gene Robinson from the University of Illinois and Sue Brown from Kansas State University. Our project is an umbrellaed association of sub-projects, the largest one currently being the USDA Ag 100 Pest Initiative to sequence the genomes of the top 100 pests in the US. We also have a USDA and University of Illinois Genome 100 project to sequence 100 species of US bees, a cornome project between my agency and the Smithsonian to sequence a dozen arthropods associated with the cornfield, a beetle genome project led at the University of Kentucky to sequence a couple hundred species of beetles, and a project in France to sequence aphids and their parasitic wasps. In addition to sequencing projects, i5K can be considered the working group that brings about the Arthropod Genomics Symposium, which, before COVID, met every year and hopes to reconvene in 2022. Please associate your project with i5K and send a representative to our steering committee. The Darwin Tree of Life project is in this way represented on our committee. The associated projects are varied in their place in the genomics pipeline, collecting samples and vouchering species, extracting DNA, doing long-read sequencing, and assembling genomes. We also maintain a funded workspace, the i5K workspace at the USDA National Agricultural Library in Beltsville, Maryland, which serves as a community hub for annotation. So why associate with i5K? Join us if you want to be a part of a team that work with partners to sequence the desert locust, an insect ravaging crops from the Horn of Africa to Pakistan. Sequencing this genome of 8.7 megabase, we were able to get chromosomal level assembly. Our members continue to push the edge of sequencing and assembly technology. We now have over 100 such successes. Join us if you want to develop grant proposals in the areas of your interest. Join us if you want to set agendas and develop sessions for major arthropod genomics meetings such as this one, or participate in webinars on new technologies and sequencing efforts. Join us to increase our diversity internationally in the taxa that we sequence in every way. But mostly join us to explore, to understand when locusts swarm, can we stop locust plagues? Can we use invertebrates as sentinels? For example, bees for climate change or amphipods for water quality. Can we protect our arthropod fauna? Bees and perhaps many arthropods are declining around the world. What do we know of their robustness and genetic resilience? Can we use that knowledge to protect species at risk of extinction? Can we use comparative genomics to crack some very fundamental evolutionary riddles? What is the stuff that natural selection works on? The singer or the song, or the network of both, or as some have recently postulated? And ticks, contenders with mosquitoes as the worst pest of man and animals, we continue to fail to protect ourselves from them. Can we use gene drives based on genomics knowledge to control them? Such questions energize our efforts. Our focus, the arthropods, is one of the largest and most diverse groups on Earth. The opportunities for discovery are endless, but your time is finite. Please join us so that we can use our cumulative time and wisdom to explore the world of arthropod genomics. Please contact any of the members or write to us at the i5K workspace at NAL. Help us advance genomics research. Come and share your advances in sequence technology, genome assembly, and comparative genomics. Together, let's push arthropods to the forefront of the genomics community.